What do rodent-borne lung infections, tick-transmitted hemorrhagic fever, and some of the world's most dangerous emerging viruses have in common? They all belong to one of the largest groups of RNA viruses, the order Bunyavirales. The order Bunyavirales contains a diverse collection of viruses capable of infecting humans, animals, plants, and arthropods. Many members are zoonotic pathogens, meaning they circulate naturally in animals before occasionally infecting humans. Several bunyaviruses have become major public health concerns because of their ability to cause severe disease and emerge in new geographic regions. Viruses within bunyavirales are generally enveloped viruses with a segmented negative sense or ambisense single-stranded RNA genome. Most possess three genome segments known as large, medium, and small segments. This segmented organization allows genetic reassortment when different viral strains infect the same host. As a result, new viral variants can occasionally emerge. Historically, these viruses were grouped within the family Bunyaviridae. However, advances in molecular virology led to reclassification into the modern order Bunyavirales, which now includes multiple viral families. Among the most important human pathogens are hantaviruses. Unlike many other bunyaviruses, hantaviruses are not transmitted by insects or ticks. Instead, they are maintained in rodent populations. Humans usually become infected by inhaling aerosolized particles contaminated with rodent urine, feces, or saliva. Hantavirus infections can cause two major clinical syndromes. In Europe and Asia, several hantaviruses produce hemorrhagic fever with renal syndrome, characterized by fever, vascular leakage, and kidney injury. In the Americas, certain hantaviruses cause hantavirus pulmonary syndrome, a severe disease involving rapid fluid accumulation in the lungs and potentially life-threatening respiratory failure. Another medically important member of Bunyavirales is Crimean Congo hemorrhagic fever virus. This virus is transmitted primarily by Hyaloma ticks and circulates among livestock and wildlife. Human infection may occur through tick bites or contact with infected animal blood and tissues. Crimean Congo hemorrhagic fever can cause severe hemorrhagic disease with significant mortality rates. The order also includes Rift Valley fever virus, a mosquito-borne pathogen affecting both humans and livestock. Outbreaks can result in substantial economic losses due to disease in sheep, cattle, goats, and other farm animals. Most human infections are mild, but severe cases may involve hemorrhage, encephalitis, or vision loss. Bunyaviruses demonstrate remarkable ecological diversity. Some rely on rodents as reservoirs. Others depend on mosquitoes, ticks, or other arthropods for transmission. This ecological flexibility has contributed to their widespread global distribution. After infection, many bunyaviruses target endothelial cells lining blood vessels. Damage to these cells can increase vascular permeability and contribute to fluid leakage, shock, and hemorrhagic manifestations. However, disease severity varies substantially among different viruses within the order. Diagnosis often requires specialized laboratory testing, including molecular detection of viral RNA and serological analysis. Because many bunyavirus infections initially resemble other febrile illnesses, accurate diagnosis can be challenging during outbreaks. Treatment for most bunyavirus infections is primarily supportive. For some diseases, early intensive medical care significantly improves survival. Research continues into antiviral therapies and vaccine development for several members of the order. Preventive strategies depend on the specific virus involved. Rodent control helps reduce hantavirus transmission. Tick avoidance reduces risk of Crimean Congo hemorrhagic fever. Mosquito control programs are important for preventing Rift Valley fever and related infections. Today, the order Bunyavirales remains one of the most important sources of emerging infectious diseases, illustrating how environmental change, wildlife ecology, and human behavior can influence viral emergence. The Bunyavirales order demonstrates how viruses can exploit diverse animal reservoirs and transmission routes to emerge as significant public health threats. Understanding these viruses is essential for predicting outbreaks, improving surveillance, and preparing for future emerging infectious diseases.